video is going to detail the body's response to disease-causing pathogens. So let's say we have this pathogen that we can represent using the letter P. And it enters into our body. So what happens is that there's a specific type of phagocyte known as a macrophage. So let's call it an M. And it will perform phagocytosis and essentially engulf this pathogen. So phagocytosis is just endocytosis, and it's the process of engulfing something. So in this case, it's going to be a pathogen, and this macrophage will partially engulf this pathogen. It will partially digest it, but it's important to note that it only partially digests it, and since it's incomplete, some of the proteins or antigens that are attached to this pathogen are still going to be present. And it's important to note that these antigens are attached onto the cell membrane or the plasma cell membrane of the pathogen. And what will happen is the macrophage will actually display one of the proteins of the pathogen cell membrane on itself. And so it will essentially present the antigen. So what we have is basically the macrophage partially engulfs the pathogen, and it displays on the surface of the macrophage a portion of the antigen of the pathogen, the antigen's cell membrane. And then this macrophage will travel to a lymph node. And what will happen is that it will present this antigen to a helper T cell. So helper T cells are leukocytes, which are just white blood cells, and more specifically, lymphocytes. This is a subcategory of a leukocyte. And the helper T cell, it has this kind of receptor. So it's able to receive this antigen that has been presented on the surface of a macrophage. And since it's non-specific, it's actually able to bind to this antigen that came from the cell membrane or the plasma membrane of this pathogen we have here. And so it'll bind to this antigen, and it will then send a signal to two other cells. Firstly, it'll send a signal to B cells or B lymphocytes. And secondly, it'll also send a signal to cytotoxic T cells. So let's represent this as a B for the B lymphocytes and a C for the cytotoxic T cells. So what will happen is that these B cells what will they do? They will form plasma cells. Plasma cells. And they will produce antibodies, specific antibodies. So, you have specific antibodies that are going to be screened, or it's basically clonal selection. It's similar to clonal selection for the creation of monoclonal antibodies, except in this case we have plasma cells which are producing specific antibodies to target this antigen. So the helper T cells essentially make a blueprint. It's almost like a lock and key. There's a specific antibody that's produced by the plasma cells generated by B cells, which are activated by helper T cells, that correspond in sort of like a lock and key for the antibodies to correspond with the antigen. So the helper T cell is going to send this kind of blueprint in order to ensure that these antibodies will correspond to the antigen that is causing disease within the immune system and posing a threat to the immune system. So the cytotoxic T cells, on the other hand, is actually going to target this guy, and target the infected cells and kill them. So they're basically killer cells. So the plasma cells are also able to become memory B cells. And what these memory B cells do is that they hold imprint of the antigen. So it basically memorizes, hence the name, memory B cells. It holds an imprint of the antigen and essentially memorizes the antigen. So in the future, if the immune system is exposed to this same kind of antigen, or the same kind of pathogen, or another pathogen holding the same antigen, your immune system will already be assimilated to the antigen and will be able to generate antibodies to prevent this antigen from wreaking havoc in your immune system. And so this is the basis for immunity. So it's also important to note that for the B cells, there's a large number of antibodies that are going to be produced as a result of this. You have plasma cells and then a large number of antibodies, and they're able to circulate in a fairly free manner. So to recap all of this, we have an invader, a pathogen, a disease-causing pathogen. It has lots of different antigens. And we have this phagocyte called a macrophage that partially digests it and then presents a part of the antigen on the surface, a part of the antigen that came from the plasma membrane of the pathogen. So what will happen from this is that the macrophage will move to the lymph node, and then this helper T cell will be able to bind to the antigen because it's non-specific. And so it'll activate the helper T cell. So after the activation of the helper T cell, it will send a signal to B cells and cytotoxic T cells. And the B cells are also known as the lymphocytes. So after this signal is sent, these B cells can become activated and they will form plasma cells and memory B cells. So plasma cells produce specific antibodies to target this this antigen that's created, that a blueprint has been created for by the helper T cells. And then the memory B cells are able to have an imprint of this antigen right here, and it's able to memorize it. So in the future, if the body faces the same kind of antigen, it'll develop some level of immunity towards it. It usually takes 2 or more times to develop immunity. And then there's also cytotoxic T cells, which are direct T cells that are able to kill the infected agent. So you can see here, it'll actually go towards the pathogen and it'll kill it. And essentially that's the process of removing some targeted pathogen from the body, or getting a successful immune response towards an invader.